All right, good afternoon, everyone. Thank you so much for tuning in and joining us today. I'm delighted to be here, and I really appreciate your time and attention. I'm Dr. Andrea Prinzi. I'm a medical microbiologist and a medical science liaison on the U.S. Medical Affairs team here at BMRU. And today I'm presenting antimicrobial susceptibility testing breakpoint updates, background, and current landscape. I'd like to note that this webinar is one of 4 in an educational webinar series provided by BMRU. There will be a series that occur after this one from some of my colleagues in marketing and medical affairs, and I really recommend that you do check those out. They're going to have a ton of really useful information, particularly if you're working through breakpoint updates. If you're watching this recorded, we just recommend that you access these according to need, but would really recommend looking at these from 1 through 4, just that'd probably be the most use to you. Today, what I'd like to do is walk through a scoping overview of the process of how breakpoints are determined and updating breakpoints, and then walk through the regulatory process for breakpoint updates in the United States, particularly because this can be really, really confusing and pretty challenging, especially for those of us that have worked or do work in the clinical laboratory. Then I want to touch on the differences between verification and validation studies, briefly touch on what the College of American Pathologists, or CAP, might be looking for during an inspection, touch on some frequently asked questions, and then take some questions and do a Q&A period at the end. The objectives for today would be that overall you understand how breakpoints are determined for different bug-drug combinations, that you are able to discuss the need to update breakpoints and why this is so important for public health, explain the difference between verification and validation studies, and then finally understand CAP requirements regarding these breakpoint updates. So let's start at what is essentially the very beginning of things when a new drug enters development and clinical trials are being done on this drug. The purpose of these trials would be to determine how this antibiotic impacts an organism, and then also what the impact is on the patient in clinical care. And during this process, tons of isolates are tested using antimicrobial susceptibility test methods, and that generates what's called a minimum inhibitory concentration, or MIC distribution. And that's what you see in the figure down at the bottom of this slide here with this blue bar graph. This is an MIC distribution, and typically for most bug drug combinations, this will follow a relatively normal distribution, or sort of a bell curve, where the majority of isolates fall sort of in the middle there. Some of them will be a bit more susceptible, some will be a little more resistant. What happens after all of these MIC data are generated is they can be plugged into a calculator, and that will calculate what's called an epidemiological cutoff value. And what this value does is it divides this distribution that you see into wild type isolates versus those that may have required some sort of resistance. So in this example here, you see that red dotted line there is showing us where the epidemiologic cutoff value is indicated. And the isolates to the left of that are probably the wild type isolates, more representative of wild type isolates. And the ones on the right have likely acquired some sort of resistance. And at first look, you may think, well, this is a great place to pick a breakpoint. But what's really important to know is that these epidemiological cutoff values do not take into account any sort of clinical information. So clinical data that were collected during trials, such as pharmacokinetic and pharmacodynamic data, clinical outcomes data in the patients in which this antibiotic was used. And so that's really important to consider because it will impact how these drugs might be dosed and the impact that they might then have on an organism causing an infection. And so these data have to be combined to ultimately determine a breakpoint. So once you combine that ECOF value and clinical data, you end up with a breakpoint, which is basically a translator for these minimum inhibitory concentrations that we get from our susceptibility testing methods. So before we have a breakpoint, this MIC is just a number. It doesn't really mean much to us. But when we apply interpretive criteria or breakpoints, then we know how we most likely should be using the antimicrobial in clinical practice. And that really helps clinicians decide how to best treat their patient. The interpretations that we often get by application of these breakpoints are susceptible, intermediate, and resistant. That susceptible category tells us that the use of this antibiotic is most likely to result in therapeutic success. The resistant category tells us that there's less likely to be therapeutic success if this antibiotic is used against this organism. And that intermediate category is a bit more complicated. This is this kind of gray zone that captures technical variability and area of uncertainty, and also includes the susceptible dose-dependent category, which is more of a clinical measure and depends a lot on different dosing regimens and things. And not going to get into that much today, but these interpretations are really what help drive selection of antimicrobials and how they're used clinically. So after FDA sets breakpoints and this drug starts getting used in the real world clinical data start rolling in. And we learned very quickly that perhaps maybe some of these breakpoints are working or maybe they're not. This is a great table that comes from a publication written by Dr. Romney Humphreys and team. It's in Journal of Clinical Microbiology from 2019. And this lists a variety of examples that may lead organizations like Clinical and Laboratory Standards Institute or CLSI to update breakpoints. So that top example there is one we're all probably very familiar with. This is the recognition of a new resistance mechanism. This is what we really saw with the carbapenems for Enterobacterales and really led to a lot of significant breakpoint changes. Other reasons may include things like there's new pharmacokinetic or pharmacodynamic data indicating that a breakpoint is too high or too low. Or another example would be that data have rolled in that perhaps the breakpoint's a poor predictor of clinical response using the existing breakpoints, and they need to be updated. So as you can see, there's a lot of important clinical impact here with the use of breakpoints. And so it may seem like because the appropriate use of these is important, that every laboratory would be using the most up-to-date breakpoints that are available to us. But I think we all know that that is really not the case. And there's a wonderful publication that came out that really drove this point home. This was published in Open Forum Infectious Diseases in 2022 and was led by Drs. Patricia Simner and Romney Humphreys and a fabulous team of individuals that are within regulatory agencies, infectious diseases, medical microbiology. It's really a wonderful paper if you haven't had the chance to look at it. And this paper really encouraged, I think, a lot of us to move and move towards action on this breakpoints challenge. What happened here was individuals from the College of American Pathologists, or CAP, had noticed that when laboratories were sending in the results from routine proficiency surveys that were sent out to laboratories, that if you've worked in a clinical lab or if in lab now, you routinely get these surveys to test proficiency, they had noticed that it seemed many labs were perhaps using obsolete breakpoints on their antimicrobial susceptibility test methods. And so to dive a bit deeper into this, they sent out an additional survey along with a routine proficiency survey to ask laboratories what they were using. And the findings from this study really helped paint a landscape of the current need to update these breakpoints, and it was actually pretty surprising. What they found was that about 55% of labs in the United States reported ongoing use of obsolete breakpoints with no current revisions in progress, and about 60% of labs reported that internationally. And while the reasons for this were pretty variable across labs, the most commonly reported reason for this was related to manufacturer issues. So 50% of labs in the United States reported manufacturer-related issues or barriers to implementation of new breakpoints, and 62% of international laboratories. And so I think this was a really big call to action for many of us in microbiology, infectious diseases industry, and otherwise, and was really striking how much of a need there is. I think we realized that labs might be a bit behind, but this paper really helped bring that into focus and demonstrate how important it is that we move on this. But updating breakpoints and the breakpoint process is very, very complicated. And I want to spend some time walking through the regulatory environment for breakpoint updates, because I think having been a medical laboratory scientist for many years myself, this was something that was not familiar to me until much later in my career. But it's very important for laboratories to understand this as it definitely helps in planning with some of your validation processes and things as you move through this. Again, this is a wonderful figure from a publication from Dr. Romney Humphreys published in the Journal of Clinical Microbiology. And I just want to walk through this starting from the left. So if you look at that new drug, that green box there, new drug, that's what I was talking about on a previous slide, where a new drug has come to market, FDA has set breakpoints, and it's in clinical use now. And so if we move up to that next column, to the right there, we see CLSI revises breakpoint. So clinical data have come in, CLSI have evaluated these data, and they've decided this breakpoint probably needs to be updated for one reason or another. When they decide that, they write up a rationale document and submit it to the FDA for review. I think it's important to note that CLSI and FDA will work independently of each other for the most part when it comes to breakpoints. So CLSI will continue to publish the updated breakpoints that they decide on in that M100 document that comes out every year, regardless of whether or not FDA wants to adopt these breakpoints. That's important to know. But the point here of submitting this rationale document to the FDA would hopefully be to align on some of these breakpoints so that we're all sort of using the same language. So once this rationale document is submitted to the FDA, FDA will review it, and they will either decide that they agree with the rationale and they will adopt these new CLSI breakpoint changes on their own website, or they may choose to not adopt them for one reason or another. The website that the FDA posts breakpoints to is called the Susceptibility Testing Interpretive Criteria website, or the STIC website. If you ever want to access this, you just can Google STIC FDA or FDA STIC, and you should be able to find this website pretty easily. So I'm going to show you a couple of examples of when something would be accepted by FDA or not. So this first example here, we're looking at a screenshot from the FDA STIC site. This shows ampicillin and the Enterobacterales. And you see that FDA has agreed to adopt the breakpoint changes. And so next to that green arrow is a little comment that says M100 standard is recognized. So that means that FDA agrees with the rationale, they've adopted the CLSI breakpoints, and the CLSI and FDA breakpoints are the same. This example is where FDA has not agreed to adopt those breakpoints for whatever reason, and they are going to maintain their own breakpoints on the STIC website. So in this case, you don't see that M100 comment, you see breakpoints listed individually here for Enterobacterales and cefepime. And laboratories can choose to use CLSI or FDA, but they will need to indicate which they're using and where they got them from. And so we're going to talk more about that in a bit, but it's important to note that the CLSI M100 breakpoints will be different in this case than what's posted on the STIC website. You can use either, but you have to be able to explain what you're doing. Again, we'll touch on this a bit more in some future slides. So what does all this agreement and everything mean for clinical laboratories and for industry or manufacturer partners? The first thing that can happen is if CLSI submits breakpoint changes to FDA and FDA agrees to adopt them, then the manufacturer can go ahead and try to seek clearance for those new breakpoints. What's really important for clinical laboratories to know is that manufacturers of automated susceptibility test systems have to follow the FDA. So unfortunately, if CLSI updates some breakpoints and FDA does not adopt them, the manufacturer cannot seek clearance for those new CLSI breakpoints. They have to do whatever FDA is doing. However, if FDA does adopt those CLSI breakpoints, the manufacturer can then pursue clearance of those new breakpoints. The first thing the manufacturer will probably have to consider is if their card or panel has a range that can accommodate those new breakpoints. If the card or panel does not, then they have to reformulate the drug and the panel. That takes a considerable amount of time and resources, and so there will be a time delay there. And then additionally, submitting data to the FDA for clearance does take a very long time in general. So even if all goes well and a manufacturer starts to seek clearance not long after CLSI has updated some breakpoints, that clearance process does take quite a bit of time. There's a lot going on at the FDA. There was a lot of impact after COVID-19. And so in general, this process does take a while. I think it's just important to know, too, that manufacturers are often juggling different priorities, different levels of resource and time commitment. So what's in the pipeline will depend on that as well. So this leads to the discussion of verification and validation. So if a manufacturer is unable to seek clearance for a set of breakpoints, then the laboratory might want to use CLSI breakpoints that are not cleared on their system, and that may lead to what's called an off-label validation. If the laboratory brings in a new card and it's got a drug that was FDA cleared on it, and then some breakpoints changed and the breakpoint changes are cleared, that may involve just a minor verification. I think there's a lot of confusion around the terms verification and validation, and you see them defined a bit differently across various things in the literature and recommendation documents. It's important to make sure you understand the difference because the workload associated with these is quite different. For verification, the CLSI M52 document provides a lot of guidance. This is a really wonderful document if you are doing a verification. That documentation will walk you through a comprehensive verification versus a limited verification, different instances of when you would do those things, and helps provide some minimum guidance on what you might want to include in a verification protocol. Most importantly, verifications are considered on-label and a bit less rigorous than a validation study. This is when something has been FDA cleared and the laboratory needs to demonstrate that it performs about as well as it did during that FDA clearance process. So laboratories are different. There's different levels of clinical and technical expertise at each setting. And so this is really just ensuring that when the test is in your hands, it performs in a similar way to how it was cleared. That doesn't really require a whole lot. It's still work, but in general, CLSI will recommend something like 10 isolates, generally that are all comers of organisms, so organisms that are representative of the local epidemiology or population at your institution. Comparator methods should be used to run this study. And then you would assess categorical agreement and assess errors as listed here. Again, what this protocol looks like will definitely change from site to site, and CLSI helps provide a lot of guidance here, but ultimately it's up to the laboratory director to determine what this protocol will look like. Validations get a lot more complicated, if you will. Validation is typically reserved for a couple different scenarios. There's a couple I won't touch on today, but the one that's most relevant for this talk is when there are breakpoints that are going to be used on a system, but they have not been cleared for that system. Since there really wasn't an FDA clearance process, the burden then sort of lands on the laboratory to really demonstrate the performance of those breakpoints, not only in their setting, but on the system as a whole. And so this is considered modification of an FDA cleared test and is considered off-label use. And so it does require more work on the laboratory's part to ensure that these breakpoints are acceptable on their system. I will note, if you're a clinician and you're watching this talk, please take a little mental note that validations are pretty expensive and labor intensive for laboratories. It's not as simple as simply flipping a switch and turning on new breakpoints on a system. There's a lot of work that goes into this, a lot that's required at the laboratory. And so working closely with your laboratory and understanding what these processes look like may be really helpful in supporting them through this process as they move forward. The example here is pulled together from the CLSI M52 document, as well as some webinars that have been provided by CLSI, which have been really, really wonderful. I highly recommend, if you're working on developing a validation protocol, that you access the resources CLSI has available. They've offered lots of great examples of how one might think through development of one of these protocols. In general, CLSI will recommend 20 to 30, but ideally 30 or more isolates, making sure that those are, again, representative of the local epidemiology, using a comparator method for testing, and then the error measurement and categorical agreement assessment will be a bit more stringent here, given that this is a larger scale validation and it's really testing the performance as a modification of the FDA cleared test. I will make a note that the CLSI M52 document is tremendously helpful. It is a wonderful resource. I think that it's great to access. It was not written specifically for validations. It really emphasizes verifications, and CLSI, I think, is working on writing a newer version of the M52. But there are a couple pages in there that are helpful for guidance with validations if laboratories want to access that. So just know that the majority of the M52 focuses on verification, but there certainly is some guidance in there for validation. Now, I just want to touch briefly on what CAP is looking for during an inspection. And I want to start this section by saying that I am absolutely not speaking on behalf of CAP. I have been fortunate enough to work with colleagues that do work with CAP or work for CAP and do a lot of really wonderful work there. And so it's been wonderful to understand their perspective and provide a little context into why they are doing what they're doing with this initiative. I think I would start by saying that CAP certainly did not do this to cause pain and suffering to everyone that's working in a clinical laboratory right now. I know that this is a terribly overwhelming process. It can be very daunting, and it certainly will require a lot of work for many laboratories, particularly in the United States. But this is a very, very important initiative. So we are in the middle of an antimicrobial resistance crisis. It's something that we can no longer afford to not completely fight in the best ways we're able to. And this initiative really helps us do that, helps us get in line with the best way to address this problem. As a clinical laboratory and medical microbiology community, it's really our responsibility to ensure that the antimicrobial susceptibility test data we're releasing are as accurate as possible, because they have direct impacts on patient care and on public health. So not only do they help guide the treatment that a clinician ends up using to manage a patient, but they also feed into things like AMR surveillance for public health. That directly impacts public health interventions for infection prevention, things like that. So this work that's done by the laboratory is exceptionally important. And the best thing we can do is make sure that we get these breakpoints up to date. And so the steps now are going to involve making sure that laboratories have the support that they need, and that we do this together to accomplish this really big goal. I want to touch on the two checklist items that I think everybody's pretty familiar with now, and just walk through those, what CAP is asking of folks, and then touch a bit more on what they might look for. So this first item is the revised criteria listed at the top there. There's the checklist number. And this essentially states that laboratories must know which breakpoints are in use at their institution. Again, laboratories can use CLSI, FDA, or UCAST. They just have to know which they are using and be able to document that. And then this must be reviewed and documented annually. So I know that there's a lot of work happening now to get up to date, but it's important to remember that this will be a continuous iterative process that will need to be really revisited every year to make sure that folks are remaining within compliance, and that we are reporting things in the best way possible for public health. Also, laboratories should discuss breakpoints in use with their antimicrobial stewardship or clinical team as appropriate. This next requirement is the newer one, and this is the one I think that everyone's pretty aware of. This states that effective January 1st, 2024, the laboratory must be using current breakpoints for interpretation of MICs, and that obsolete breakpoints can no longer be used. An obsolete breakpoint would be a breakpoint that is not currently published on either that FDA STIC site or published by CLSI or UCAST. So again, there can be a combination of these breakpoints in use in a particular setting, as long as everyone is documenting what they're using and what their plan is to update. That will be very, very important. So no obsolete breakpoints as of January 1st, 2024. I was lucky enough last year to talk to Dr. Carol Roche, who works really closely with CAP and is very involved in a lot of these initiatives, about the mission of CAP and the things that they're working on related to this particular initiative. And really some of the bottom line takeaways I got from that conversation, which we wrote up in an article that was published in ASCLS today, and I'm happy to share with folks if you're interested in reading that. But essentially, CAP inspectors do not require any specific format for these verification or validation protocols, so long as the documentation is clear on what was done and what was found, as well as these annual reviews of data and which breakpoints are in use. You should really be using resources from CLSI, CAP, other organizations to help guide you. But again, what these protocols and documentation types look like will be up to each laboratory director, manager, supervisor, whomever is responsible for putting these things together. And a lot of discretion will be afforded to those folks regarding validation protocol details. Each site, really, it's recommended that each site determines their own level of risk and need and then rates the protocols up appropriately. But there are a lot of resources out there to help folks from our wonderful colleagues at CLSI and otherwise. I wanted to just briefly touch on some frequently asked questions that we see, just to kind of put some thought into these, but of course I'm happy to elaborate on these or answer more during our Q&A period. The first one I've seen come up a bit in some CLSI webinars and things too, and it says, since application of new breakpoints does not change the actual MIC or the original MIC, and since we're only assessing categorical agreement, can we just rerun old data? And CLSI talks a lot about the use of a comparator method and how important that is for assessing performance of your new breakpoints. If you're simply comparing old data to new data, the performance is probably not going to look great. The agreement will probably be poor. And so CLSI is not really recommending that be a method that is used. I think it's really important to think about performance of drugs like piperacillin and tazobactam, where the new breakpoint cuts into the MIC distribution. So there's a lot less room for error there. There's less room for variability. And so a little bit of movement could throw the agreement off a bit. So really a reliable comparator method is what should be used according to a lot of the resources that are out there in order to perform these verifications and validations. And that leads to the next question, which is essentially, do we have to use a comparator method? And the answer is yes. A couple things I'd like to know here. This is a screenshot of the CDC AR isolate bank. If you have not accessed these yet, this is really a great resource that's available to clinical laboratories who are performing validations. These are panels of well-characterized organisms that can be used to run on the system and perform a validation. This is accessible through the website. Panels can be ordered there. And again, it's recommended you refer to the CLSI M52 document for specific guidance around development of that protocol. And then questions come up quite a bit about how many isolates should be tested for a validation or a verification. And I think I'm repeating myself over and over here, but this will really depend on local needs and preferences. I've talked to some clinical sites where 30 isolates is not considered acceptable for a validation. They want to see maybe 100 isolates or more for a given bug-drug combo. This is really dependent on the needs of the local setting, the risk that's determined to be present, and what is feasible given resources and tech time in particular. In general, CLSI is recommending around 20 to 30, but probably 30 or more isolates per antimicrobial agent with high and low MICs for those agents that are being tested. Again, it's recommended that whatever you're testing is representative of what you typically see in your hospital setting or at your laboratory. And there's, again, a lot of really great resources about both validation and verification in the CLSI M52 documents and on the webinars that they have posted recently, particularly the last CLSI M100 document update webinar was exceptionally helpful for these kinds of things. So with that, I will stop going through slides and open up the floor for a question and answer session. And then just thank you so much for your time.
Well, thank you very much, Andrea, for that great presentation and setting us up for a very interesting and invigorating discussion throughout the rest of the day. The questions are rolling in, so we're going to go through some of those. But to everyone out there, please keep them coming in. If we don't get to them by the end of this session, we do have your email, and we will be able to follow up with you directly. So that we can hopefully get through most of the questions, we'll jump right into the first one here. So are there any other resources coming out or that are currently available from organizations like bioMérieux or the CLSI that will help labs get through the breakpoint update process?
I love this question. Yes, so there is a lot happening. I'll speak to bioMérieux first. I can say I'm really, really proud of the work that's been happening internally here at bioMérieux between our medical affairs group, as well as our marketing colleagues and our field application specialists, all of these folks that are in the field engaging with our laboratories. We've been doing a lot of work on internal and external education, developing different tools and resources that can be used by customers, laboratorians that are going through validations. And I think the webinars that are coming up are going to touch on some of these wonderful things. So I'm not going to elaborate on those too much. But yes, a lot of work is being done internally here. We're very, very passionate about making sure that we get up to speed on this and that we do what's best for patient care. I also want to compliment our wonderful colleagues at CLSI, APHL, ASM, CAP. These folks are all working together really closely to come up with a variety of things that I think will be wonderful for laboratories. The first one that I think is coming any day now, I hope, is being developed by CLSI in collaboration with all those other folks I mentioned, called the BIT, I think. I think it's the Breakpoints implementation toolkit. This was mentioned at the most recent CLSI update webinar. And they did say this would be coming out in the spring. And I know it's going to include, I think, things like organism sets available through that CDC AR bank, sets that maybe are a little more conducive to some of these validations that are needed, educational materials, resources, things like that. I know that CAP's been doing a lot of educational work in both in real-time presentations as well as written works, and then organizations like ASM are doing a lot of science communication and advocacy work around this issue as well.
Yeah, thank you so much for sharing all of that with us. We'll keep moving with our next question here. So what do you think is needed to make this process more streamlined in the future?
So I think that we have really seen in this particular challenging, complex situation that communication and collaboration is really the name of the game here. I think historically we've seen that our sectors are all a bit siloed. So between clinical laboratories, public health, industry, clinical practice, things are always a bit siloed. And so we don't always understand the needs or abilities of each of these areas and aren't always able to address them as readily as maybe we'd like. And so what's been really wonderful through this process has been a lot of the collaborations that are starting to occur between all these agencies, and recognizing that we can all help each other get through this a lot better, I think, if we communicate. So I think in the future, plans for a lot more communication and understanding the needs. I think from the industry perspective, we're really interested in being innovative, thinking outside the box, and doing everything we can to make sure we're addressing an urgent public health need, supporting all of our clinical public health partners, doing what we can to make sure that the tests we have are addressing those needs as well. So I think collaboration and transparency is going to be really key.
Yeah, I love to hear about communication, collaboration, really helping us, reach across to sort of all different paths and helping to make that work better for our patients. Yeah. So you've talked a little bit about this, and wondered if you could just expand, maybe a touch and direct to some future discussions we'll be having. But is bioMérieux going to be providing any kind of tool to help customers determine what's going to be needed to be validated for some newer panels that may be coming?
Yes, there's been a lot of work for quite a while now on the development of sort of an audit tool that will help customers go through what they're using on their system, identify which breakpoints are already being used, understanding what has been cleared on each of their cards, and then determining whether or not they would need to do a verification or validation. I think it's important to note, too, that we have a really nice medical affairs infrastructure within bioMérieux that's been really wonderful. The folks on this team are infectious disease pharmacists, Ph.D. level clinical microbiologists, other types of clinicians that have been in clinical practice recently. And so it's been really wonderful to get engaged with this project and help people think through some of these issues on a deeper level, because it gets so messy and complicated. So U.S. Medical Affairs has been really involved in development of that tool as well. Our phenomenal marketing colleagues also working on that. So yeah, that will be hopefully something that can be used to identify where the gaps are and then make a plan for addressing them.
Perfect. And we certainly hope that tool is very helpful. So if we could pivot a little bit and talk about, so when folks are going to be doing these validations or verifications of their new breakpoints, potentially with, excuse me, or without that tool, there's a question asking to clarify, so is it not okay to use clinical isolates for establishing the revised breakpoints? So more specifically, do we have to use the CDC AR bank organisms?
No, nobody requires that you have to use those. I think that option is most reasonable or feasible for labs because if you don't have another automated susceptibility test system in-house that's been validated with the new breakpoints, it could be hard to use your clinical isolates as the things that you're testing. Why labs often choose to use the AR bank is because they are already well characterized and their MICs and everything are already known. And so you have that information, then you run it on your system and you can compare what you get on your system to what's already known about these organisms. So it's not like you have to have another instrument in place there. But certainly if you have a reliable comparator in-house, you can run a combination of your own clinical isolates as well as QC isolates, and that is acceptable on CLSI states. That's totally fine.
Perfect. Thank you so much for hopefully clearing that up there. So a bit of a tricky question here, short but tricky. So how long do, so as we were talking about the review process, walking us through as a new drug comes to market and then it goes through the whole process with that lovely flow chart, how long do FDAs ultimately tend to take?
This varies. Again, nothing in this space is black or white. There's lots of gray area here, and I certainly don't want to speak for the FDA. But I have seen and do know that some of these have taken 7 months or longer for some of these clearances, and I know that it could be even a bit longer post-COVID. So anywhere between 7 to 12 months, maybe shorter in some instances, but many months, we will say many months typically.
Yeah, I think there's a lot of variability, right, when it comes to the regulatory landscape, and there's a lot of moving parts to it.
Yeah.
So sticking in sort of a similar vein about clearance of items and package inserts. So for AST cards that have a package insert indication for reporting drugs with select organisms, so if an institution wanted to implement a card and wanted to validate an organism, but there wasn't a specific listing for a given drug, would a site have to test 30 isolates of that same organism?
So I am going to say first, there's a caveat to this. This kind of questioning is best directed towards either your strictest regulatory agency or to, if you have access to a medical affairs department with the industry partner you're working with, they would be the best folks to help walk you through this. A lot of folks are really struggling with some of the changes that have happened where at one point, organisms were cleared in a package insert as clinically significant gram-negative bacilli, as kind of this big group. And so anything that folks thought was clinically significant gram-negative bacilli probably fell in there. And now things are listed very differently in these package inserts. They are specifically listed as genus and species organisms that were cleared specifically for that use. And so conversations around what your options are definitely more of a deeper dive off-label conversation. No one can specifically tell you how many isolates you have to run. You should definitely refer to CLSI M52 for some guidance on that. But generally speaking, if it's outside of what's in the package insert, it's off-label, and so you fall into validation land there. And the requirements for the protocol are going to be a bit more strict and laborious. Probably.
Perfect. Thank you. So we have a couple of questions coming in about other methods of doing AST specifically things like disk diffusion, e-test. So could you comment first on whether disk diffusion could be used as a comparator for a validation or verification, I believe?
So I think that this one, again, would be a good one to reach out to clinical micro colleagues. I'm going to give Dr. Romy Humphreys a shout out here. She's really the leader, amazing leader in this space that can answer all of these questions right off the top of her head. I'm pretty sure that CLSI does state that disk diffusion is an acceptable comparator, although there are some drugs that are a bit more challenging when using disk diffusion as a comparator. So I think that one, it would really depend. Again, it would be a case-by-case scenario. We'd have to probably look into a bit more. Happy to dig into that a bit more with folks if needed. The most important thing is whatever comparator you're using does need to be cleared at the breakpoints you're looking at. And if you're looking at things like disk diffusion, you have to remember that the measurement for those zone sizes is not the same as an MIC, but since categorical agreement is what we're comparing, it is possible to compare those sorts of things. So I think special thought should be given to the drugs that you're looking at, the organisms you're testing, and again, reference what CLSI recommends. The M52 document, I know I keep saying it, M100 and M52, I say them so much because I use them probably 100 times a day. So M52 definitely lists out the types of comparator methods you could consider using. And is a really wonderful resource for considering those options.
Perfect. And so asking sort of a similar question, but with regard to, so if you were bringing on an e-test with a revised breakpoint, what would that potentially, or are there any considerations that need to be brought up?
Just again, your comparator method would need to be something that was validated with the new breakpoints. So if you don't have that in-house, perhaps considering sending to a reference lab, making sure that the e-test that you're putting in use has been cleared for those breakpoints. This does bring up a good point. Breakpoint updates don't only apply to automated susceptibility test systems. They do apply for manual methods as well. So reviewing all of the breakpoints that you're using across your methods is important.
Great. So for, I'm sure there are a lot of labs out there that with this talk, as you've mentioned, the number of labs that are not necessarily up to date on their, with their breakpoints, and they're thinking, oh, well, I'm going to have a lot of validations or verifications to complete to bring myself up to be 100% in compliance. So do you have any recommendations for resources that they could look at to maybe prioritize those drug-bug combinations that they're going to want to tackle first?
Yes, this is also complex. I'm sorry that I say that about every answer. There's some really, really great resources available across the board. One that comes to mind is the wonderful publication, again, by Dr. Romy Humphreys. This was, I can't remember the exact title, but it's basically the Understanding AST Breakpoint Updates. I think it's the one that says a primer for clinical laboratories. This is a really wonderful publication overall, but the supplemental documents in that publication are really, really helpful. There's one supplement in there in particular that provides sort of a layout, an algorithm for thinking through prioritization of these drugs. So there are certainly some drug-bug combos that should be validated sooner than later based on clinical and public health need, and those are laid out really well in that paper. I think also CLSI's most recent webinar touches on this as well. There are some that are more urgent than others and maybe more feasible than others. So definitely I would prioritize based on that. And then absolutely, I think working with your clinical colleagues and talking to your stewardship team about what's on formulary, what can be suppressed, what's actually needed for clinical care right now, prioritizing these options with the clinical team based on what the resources and availability of this kind of testing is in the lab is really important. I do know that there are a lot of laboratories out there that may not have a stewardship team on hand or access to a clinical team like that. So publications like the one I mentioned earlier, or resources through CLSI would be really, really wonderful for groups like that might need some guidance, but perhaps don't have access to that clinical team.
Super. I think all that is great.
Great. All right. I think we're wrapping up here. I think we're at 45 minutes. So I think we're going to have questions answered too for those that have put some in the Q&A. I think we'll follow up with those later. So just want to say thank you all so much and have a wonderful day.